introduction to the Archegoniates. So we have studied in the previous about algae and fungi and what are Archegoniates. So Archegoniates are one group of plants which contain Archegonia, the female reproductive organ. So in simple way, Archegoniates are bryophytes, pteridophytes and gymnosperms, not angiosperms. Because in angiosperms the archegonium is developed and we call it as ovary, ovule and embryo sac. Now this Archegoniates, they are the lower group of plants which include bryophytes, pteridophytes and angiosperms, gymnosperms. Now the main character itself is they contain Archegonia, female reproductive organ. Now in the previous part, when we study about fungi, we don't come across such kind of structures. Of course, there are sex organs which involve in the sexual reproduction, but we don't come across structures like Archegonia as found in bryophytes, pteridophytes and gymnosperms. So it means that they are less developed group of organisms, fungi. And even in algae also we come across antheridium and also we come across oogonium. But however, there are certain algae which also contain Archegonia. So we can also include some algae in this group of plants. So certain algae also are also included under this group of Archegoniates. So Archegoniates are compulsorily bryophytes, pteridophytes and gymnosperms, which may include certain algae. Now this algae and fungi, they are less evolved when we compare to the Archegoniates. When we say Archegoniates means it is a evolved stage. The presence of antheridium and Archegonium is a step of evolution. But in fungi and algae, we are having sex organs which are somewhat simple, not developed as in case of the Archegoniates. So in other organisms, for example, if you take virus and bacteria, there still we don't find the sex organs at all. In virus and bacteria, if you just come across, which we have already studied in the microbes, so the viruses, they are somewhat a peculiar group because they possess living and non-living characters. So living and non-living characters which are possessed by us, they show their peculiar nature. So when we say living organisms, it means that they are having only living characters, no question of non-living. But surprisingly, viruses are having living and non-living characters. So that is why some taxonomists, they do not classify virus in living organisms. They classify the viruses under some other group, separate group. They do not include under any other kingdom or any other group. If we take the example of Whitaker's classification, what we have studied in lower class. So Whitaker's classification mentions that the living organisms are classified into 5 groups: Monera, Protista, fungi, plantae, and Animalia. Now, when we observe viruses are included under which group or which kingdom, Whitaker has placed viruses in a separate group which he does not mention in any kingdom. He does not classify viruses in any of the kingdoms what he have mentioned, neither in Monera nor Protista, or even in the fungi, and never in plant and animals. So viruses are very peculiar type of organisms. So they are not made up of cells. They are only made up of protein and nucleic acids. Now, because of this absence of cells, they are considered as non-living. They are without cells, and they remain as inert particles. So hence they are considered as non-living. They can be stored in the bottles as chemicals, so they are non-living. But when we see the living characters, how they are considered as living, they reproduce inside living cells. When they are outside living cells, they remain as they remain as inert particles. But when they are inside living cells, they reproduce. So when we study the characteristics of living beings, living organisms, growth and reproduction is the important character of living organisms. But however, we don't find growth in the virus. We can see the process of reproduction in the virus. Now, because of the reproduction, we consider viruses as living, and hence they are considered as living, means they possess living characters. And not only this, because of the presence of nucleic acid, DNA, RNA, they are considered as living. So presence of nucleic acid indicates the living nature for that substance or for that material. Anywhere you find DNA or RNA, it is involved in replication, so it indicates the living character for that object or for that part. And also they respond, they are sensitive to heat and cold. If there is variation in temperature, so they respond to that high temperature and low temperature. That is why the living character of viruses is considered. So this viruses are actually a material for evolution. So when evolution took place, so where we are coming to the Archegoniates, which are highly developed group of plants. So the viruses also may have played an important role in evolution. So because before the evolution of living cells, the viruses might have been appeared on this earth. The formation of proteins, the formation of nucleic acids might have occurred, and these proteins and nucleic acids, so they have organized to form the virus particles, and gradually these viruses might have changed and given rise to living cells. So viruses are can be considered as the basis for evolution of various organisms. And then after viruses we come across bacteria. So bacteria, they are unicellular but very simple cells. They are primitive because they are prokaryotes. So when we study the bacterial cell, in a bacterial cell it is very simple. The DNA material is freely present in the center without any nuclear membrane, which we call it as the incipient nucleus, or we call it as indefinite nucleus or nucleoid. There is no mitochondria, there is no Golgi complex, there is no endoplasmic reticulum in the cells, and the cells are very simple with the ribosomes, and of course the cell wall and all that, it is present. So like that, when we come across to the lower organisms like viruses and bacteria, they are still simplified. And again, in viruses we are not having any such type of sexual and asexual reproduction. But in viruses there is a simple multiplication. It is usually referred as multiplication because after the formation of virus there is no growth of the virus. The newly formed particles are almost as that of the parental molecule, the parental viral particle, and that is why only we call it as multiplication of a virus. But in bacteria, when we see, we come across asexual and sexual reproduction. So asexual means it takes place by fission and endospore formation. Sexual reproduction, here there is no any such kind of male and female. Even then there is just a transfer of the DNA from one cell to another cell, where we call it as genetic recombination. So the bacterial recombination we call it as. So it takes place by 3 steps like conjugation, transformation and transduction. And the DNA which is transferred to the recipient cell, the DNA which is given from one cell, it is the donor cell. The cell which receives, it is the recipient cell, and thus the recipient cell acquires a new set of characters, and it is because the DNA which is donated by the donor cell, and that is just an improvement in the reproduction when we compare to the lower ones. Now in the conjugation also there is the transfer of DNA from one cell to another cell. And then in the next group, like algae, we see that the algae has both vegetative reproduction, asexual reproduction and sexual reproduction. Vegetative reproduction by just commonly fragmentation, and then asexual reproduction by the formation of spores. Various kinds of spores are produced there, and in sexual reproduction we come across isogamous, anisogamous and oogamous. Isogamous means it is a very simple type or a primitive type of sexual reproduction. In sexual reproduction, there is the presence of male and female reproductive organ, or the production of male and female gametes. There is a distinguishing of the differentiation of the male and female in the organisms, and the male produces the male gametes, the female produces the female gametes. So when the two gametes are similar morphologically in shape and size, it is isogamous. So here we cannot differentiate which is male gamete and which is female gamete. Thus there is the differentiation of the male and female is very difficult. But however, the difference is in the genetic constitution. So in the male the genetic content is one type, and in the female the genetic content is of different type. So these gametes, they differ in the genetic constitution, but in the size and appearance they are one and the same. That is why isogamous is very primitive type of sexual reproduction. But in anisogamous, there is still just an means evolutionary step there, stage, where there is a differentiation in the male and female gamete. Usually the male gamete is very small and the female gamete is large. We can differentiation which is male and which is female. In case of isogamous, it is not able to differentiate by their morphological appearance. But in anisogamous, it is easily differentiated by the size of the gametes. And again in the oogamous, here there is clear formation of the sex organs. There is a well-developed male sex organ and a well-developed female sex organ in that organism. So there is clear formation of the male sex organ, and here we refer it as the male antheridium, and the female usually in some algae it is referred as archegonium, but in most of the algae it is oogonia. Antheridia and oogonia are the reproductive structures which are present in the algae. So that means it is somewhat an highly evolved one. So when we come to the next group after algae, in fungi, which are purely heterotrophic organisms, they are achlorophyllous. Algae are chlorophyllous, they contain chlorophyll, they are photosynthetic, but fungi are saprophytic or they are parasitic. That is why here the reproductive organs are also somewhat simple. There are certain fungi, highly developed fungi, which contain the male and female sex organs, but not well developed as antheridium and archegonium. But they are very simple type of reproductive organs there. That is in case of the fungi. So now coming to this archegoniates. So just as an introduction to the archegoniates, so basically what type of organisms are present with the way of reproduction, especially with reference to the sexual reproduction. So archegoniates, as I already mentioned, they contain archegonia, include bryophytes, pteridophytes and gymnosperms. So this archegoniates are the one which include bryophytes, pteridophytes and gymnosperms. So the bryophytes are the first to be formed from algae. Some of the well-developed algae like chara and others, they might have given rise to simple bryophytes like liverworts in the course of evolution. So in bryophytes, liverworts are primitive, and whereas mosses, they are well developed, more evolved bryophytes. But bryophytes are non-vascular. They don't contain xylem and phloem. That is the basic structure or the characteristic of bryophytes. So in the bryophytes, they actually lived in water. See, liverworts, take for example Rixia, Markensia, all those. So these liverworts, they actually live in water, moist soil. And for reproduction, they require water. Though they live in soil, they live on land, that is wet land, not in water purely, like floating or like that. But they require water medium, aquatic medium for reproduction. So the gametes which are produced, they require, means for their locomotion, they require water. They require aquatic medium for their locomotion. That is why bryophytes, though they were originated in water, so that is they started shifting to the land habitat. So they evolved, they originated in water, but when they lived on land, they were dependent on water for reproduction. So that is why this plants like bryophytes, simple primitive bryophytes, which were present in water, they shifted to the land habit. So this was a kind of transition stage, which we can say. It was a transition stage for the plants, where they shifted to from water to the land. Now in these early plants, it was the presence of wax layer on the plant. So this waxy layer which was present on the plant surface, which is called as cuticle, so it prevented the loss of water and helped to conserve water or to seal the water in the plant, so that when they are present on land, they can utilize that water. But water was also essentially required for reproduction, for the movement of the gametes. Now again here to allow the plant to retain water and also bring about exchange of the gases, minute pores, which are called as the stomata, they formed in the plants, which was the next step of evolution in the transition stage. So in the transition stage where the evolution took place, there the formation of stomata appeared. So that means these are all the steps towards the formation of archegoniates. So not only the formation of archegonia is considered, associated with that or along with the formation of archegonia, all these changes took place during the transition stage where the plants lived in water and shifted to the land on which they lived. Then the stomata, they helped in the exchange of gases, which was a next step in the formation of the evolution of the archegoniates. Now when these plants lived on land after evolution, they had to face four important challenges. So that means they had to obtain the source. Now when the plants lived in water, the complete surroundings was water, so it was the only medium for these plants. But when the plant lived on land, they had to depend on the atmosphere and also on the soil. Hence, the obtaining the resources was from two sides, means it was from two places, from the atmosphere and as well as from the soil. Though it absorbed water and minerals from the soil, it had to obtain carbon dioxide from the atmosphere for photosynthesis. But in water, it was the dissolved carbon dioxide which was used by the aquatic plants. Hence here, when the plants lived on land, this was one of the major challenge for the land plants to obtain the resources from two sides. Then as the land, it was open, the plants were subjected to wind and also rain like that. So it was the subject of the environmental factors to these plants. So especially the plant has to stand erect to expose the parts to light for photosynthesis. So making the plant to stand erect or upright was another challenge for the plant. So it had to develop this mechanical tissues in its structure, or the existing tissues, they should become mechanical in function with some additional modifications, with some modifications in their structure. But in aquatic, there is no necessary of mechanical tissues. So soft tissues is okay to adapt to the aquatic life. But when the plant has to stand erect or upright, so it requires mechanical tissues, and so that was another challenge for the plant to develop the existing tissues for mechanical support. That is the second challenge. Then the plant has to maintain moisture. That is also another important challenge it has to face. Maintaining the moisture in the plant was also another challenge, and also reproduction, how it has to reproduce in that open area or the open land area. So that was also another challenge for the plants to live on the land. Then so these algae and other aquatic organisms, they acquire their sources and they need from the surrounding water. And in the archegoniates, another important feature is they exhibit alternation of generations. So when we study the unique features or the characteristic features of the archegoniates, the main character is presence of female reproductive organ archegonia and male reproductive organ antheridia is the important feature of archegoniates. So because of the presence of archegonia, female reproductive organ, it is called as archegoniates. The male is antheridium. And then these plants, they are photosynthetic. They contain chlorophyll. They are independent. They contain various kinds of chlorophyll, not only one type, chlorophyll A and chlorophyll B. Along with that, there are some orange colored pigments like carotene. So those carotene pigments, they also support the functioning of the chlorophyll. So that is the another characteristic feature of the archegoniates. Then in the life cycle of the archegoniates, we come across gametophyte and sporophyte. So gametophyte means the part of the plant which produces gametes. Sporophyte means which produces the spores. Now this gametophyte, the gametophyte are somewhat we can say, so gametophyte are haploid, whereas sporophytes are diploid. So that means the gametophyte is haploid and sporophyte is diploid. And here the sporophyte gives rise to gametophyte, gametophyte gives rise to sporophyte. Now this gametophyte produces gametes, and these gametes which are haploid, so they are produced by mitosis, and these gametes, when they fertilize, during fertilization they form the zygote. So the zygote develops into the sporophyte. So the zygote is diploid because there is fusion of two N gametes, haploid gametes. Now these sporophyte, they produce spores, Now these spores are haploid and they are produced by meiosis. These spores, they germinate to give rise to the gametophyte. This is how always a gametophyte gives rise to sporophyte, a sporophyte gives rise to gametophyte. So this is called as alternation of generation, the characteristic feature of bryophytes and also even pteridophytes. So gametophyte gives rise to sporophyte and sporophyte gives rise to gametophyte. So this alternation of generation was actually first discovered by Hofmeister in the year 1857. So this phenomenon of alternation of generation, where the gametophyte gives rise to sporophyte and the sporophyte gives rise to gametophyte, was first observed by Hofmeister. So gametophyte, it produces the gametes by only mitosis. But usually when we see the formation of gametes in the higher plants and animals, so there the meiosis takes place. So this is actually the important difference between archegoniates and the higher plants like angiosperms and even animals. So in angiosperms and animals, where there also there is sexual reproduction, there also there are sexual and male and female reproductive organs, but the way of the formation of gametes is different. There the formation occurs by meiosis, where the chromosomes are reduced. It means the plant and the animal, they are diploid and they give rise to haploid gametes. But here in archegoniates, where the alternation of generation takes place, especially in the bryophytes and pteridophytes, the plants, gametophytes, they are haploid itself. So when they are haploid, they cannot undergo meiosis. So they undergo mitosis and produce haploid gametes. But however, as usual, the haploid gametes during fertilization, they fuse to form the zygote and the zygote develops into the diploid sporophyte. But the diploid sporophyte develops spores by meiosis. There is a reduction in division. So the diploid sporophyte gives rise to haploid spores. Each spore germinates into a gametophyte. That is how actually alternation of generation takes place. This alternation of generation is also known as metagenesis. So metagenesis is another term for this, where the type of life cycle is present in plants and algae with the distinct haploid gametophytic generation, that is the sexual stage, and the diploid asexual stage. So the presence of sporophyte indicates the asexual stage, where the spores are produced with the n number of chromosomes and the sporophyte itself is diploid. So the sporophyte produces haploid spores by meiosis and the chromosomes which are reduced to half from 2n to n. The haploid spores they germinate and grow into a haploid gametophyte. And then the gametophyte produces gametes by mitosis, and then the number of the chromosomes do not change, they remain constant, that is the haploid. Now such haploid gametes, two gametes, male and female, either they are isogamous or anisogamous, like that. So whatever it is, so the two gametes, they fuse to form the diploid zygote, and this diploid zygote develops into the diploid sporophyte. So the diploid sporophyte, they gives rise to the gametophyte, and even the gametophyte gives rise to the sporophyte. So in this way, all the land plants and many algae undergo sexual reproduction in this manner. Now the next aspect is, what is the relationship between the sporophyte and gametophyte? What is the relationship? So when we see the relationship between the sporophyte, this relationship differs in various groups of plant. Among the bryophytes, and pteridophytes, and also in the gymnosperms, they differ each other. Now in the liverworts and mosses, which is a group of bryophytes, the sporophyte is not well developed. Main thing, the sporophyte is less developed than the gametophyte, and it is purely or mainly dependent on the gametophyte. So in liverworts and mosses, the sporophyte is not well developed, so it is less developed, and hence it depends on the gametophyte, and it is developed on the gametophyte itself, and it is largely dependent, in other words, it is a parasitic on the gametophyte. So to give a clear idea, the sporophyte is a parasite on the gametophyte. It is dependent on the gametophyte in case of liverworts and mosses. But in the ferns of the pteridophytes, the gametophyte is called as prothallus. There the particular word is used for the gametophyte in ferns there, which is a group of pteridophytes. So the prothallus is independent purely. Now this prothallus gives rise to sporophyte, but the sporophyte is initially dependent on the gametophyte, and only for a short period. So when the gametophyte gives rise to the sporophyte does not develop independently. For a short period, it lives on the gametophyte, and after that it remains independent for a long period in its life cycle. So during its lifespan, the sporophyte which depend on the this prothallus only for a brief period, for a short period, and then it becomes independent. So only for its nutrition, for the early development, we can say. So when the sporophyte is born from the gametophyte, so this sporophyte cannot live independently. So for a brief period, for its nutrition, it depends on the gametophyte, and further it lives independently for a long period. So in general, we can say that the sporophyte and gametophyte are independent in pteridophytes. But in bryophytes like moss and liverworts, the sporophyte is dependent on the gametophyte. It is a parasite on the gametophyte. This is what the relationship between the sporophyte and gametophyte in bryophytes and pteridophytes. But in angiosperms, what happens? So there the gametophyte is very reduced. We cannot see a distinct gametophyte there, and this gametophyte which is reduced, it is very reduced to such an extent it consists of a very few cells, and this gametophyte which is very small and which consists of a few cells, it grows inside the sporophyte. In other words, the gametophyte is parasite on the sporophyte. See, it is just opposite to the bryophyte. In the bryophyte, we had mentioned the sporophyte is dependent on the gametophyte. It is a parasite on the gametophyte. But in the flowering plants, in the higher plants, see here, the gametophyte is not well developed. It is highly reduced, consisting of a very few cells, and though it is reduced and very consisting of few cells, but it is not independent. It is completely depending on the sporophyte, so it is entirely growing inside the sporophyte. This is what actually is seen in the higher plants. So like that, the bryophytes, pteridophytes, and the higher flowering plants, they exhibit different types of relationship between the sporophyte and the gametophyte. So the gametophyte and sporophyte are seen in the archegoniates clearly, which we can differentiate into a separate part and whether they are independent or dependent, we can differentiate when we compare to the this higher flowering plants. So this bryophytes are one group of archegoniates which exhibit the relationship of the sporophyte and the bryophytes. So that is about the introduction to the archegoniates. In the next part we have to study about bryophytes, pteridophytes and gymnosperms separately with one example each, one or two examples each with their life history of those plants in the bryophytes, pteridophytes and gymnosperms.